Okay. Well, hello, good morning. Thank you for coming. It may still be early for many people. Had a last night fun. We definitely had some fun. I'm really excited about this session on AI and proteomics because we hear so much about AI these days, and really, last time I wrote code was in the middle 80s, and so I want to know what's happening and where some of it is that we've been using for years and don't know it, and where the field is going. Before we get started, I have some housekeeping notes for you. Please silence your cell phones. Photography is permitted, and unless the speaker says to you not to. Recordings of the session will be available on the meeting website after the conference. ABRF's social media policy is that everything can be shared. If a speaker asks that it not be shared, please respect the speaker and do not post anything on social media. Hashtag for this event is, what do you call it? I don't know. I don't do hashtag pound, dollar sign, no, what is it? Number sign. All right, ABRF 2025. And we're going to hold questions until the end. I think that'll work better, and we can have some general discussion then. I'm sure by this time you've downloaded the app, so don't need to tell you about that. And there will be an evaluation form coming after the conference. Please, please fill it out. Any ideas you have, things that are plus or hopefully not minus, but suggestions. And then also if you have suggestions for topics for next year, they're greatly appreciated. So we're going to have 3 people presenting today. We're going to start first with Graham Wiley from University of Oklahoma Medical Research. And he's going to give us information about Olink, who's the sponsor for this session, talking about implementing the Olink platform in a sequencing lab. Then we'll have Sebastian Paez from Talis Bio. And then finally, Justin Sanders, University of Washington. And really looking forward to hearing what you have to say. So, Graham, please. Would you do me a favor? Thank you. We'll have to be careful.
Okay, good morning. I'm not going to be talking about AI at all. And that wasn't my idea. This is just where Olink stopped me. So, do we have any NGS labs in the house? Maybe. Next generation sequencing. Do we have any sequencing labs in the house at all? No. Okay, not the target market. So bear with me and we'll get through this real quick. Okay, like chocolate and peanut butter, they asked me for a title, and I came up with this: chocolate and peanut butter. 2 things that go great together, NGS and proteomics. So, like NGS, chocolate is one of the greatest inventions of humankind, and like peanut butter, proteomics has proteins in it. We got it. Okay, disclosures. I'm here on Olink's dime, but I'm going to say a whole bunch of stuff that they have not vetted, and this is approved for general audiences. Okay, so introductions. I run the Clinical Genomics Center at Oklahoma Medical Research Foundation. We have that one. NovaSeq X Plus from Illumina, Illumina NextSeq 2000, MySeq and iSeq, and 2 Oxford Nanopore P2 Solos. We have about 120 annual users or labs per year, and about a 90% cost recovery, so we're doing all right. Little asterisk there, we could be closer to 100 if the CFO would stop expecting me to cover the lease payments for the NovaSeq X, which I think should count as capital equipment, but we can talk about that later. Okay, so proteomics in my genomics core. So we have to sort of think about what the great core director Jane Austen said in Pride and Prejudice and Funding. So it is a truth universally acknowledged that a core in possession of a NovaSeq X must be in want of projects. So basically, I'm not going to sugarcoat it. This thing is a beast, and I want to cram as much stuff into the beast's maw as I can, as long as somebody will pay for it. Okay? So, Olink. Olink is actually a really interesting technology, and they're not paying me to say that. It is a really interesting technology. Okay, so the basis of Olink is this PEA system down here, wherein we have two paired antibodies that are each tagged with complementary oligos of DNA. And so when they each bind to a different epitope on the same protein, those pieces of DNA will anneal, and then you can extend, and then you can PCR, amplify that, and basically just throw that PCR amplicon onto a sequencer, and then just count how many times you see the different codes. So what's really, really cool about this, not only that, you know, it just works, is the fact that we can do 5,300 proteins assayed at a relative quantitative level at the same time with 2 microliters of input. Now you gotta have 10 microliters just as dead volume in the assay, so the robots aren't, you know, pipetting air. But we ask people for 10 microliters, and we take 2 microliters, and then we can give 8 microliters back, which is really attractive to people who have really, you know, precious longitudinal samples. So we decided that it was really interesting. We decided there was a market. So we became an Olink certified service provider. So that's us there on the Olink CSP's webpage. So to start off with Olink Explore HT, which is the major platform we're running, we've got to do a little bit of mise en place. All right. So to start off, you absolutely have to have an SPT LabTech Dragonfly. You absolutely have to have either a SPT LabTech Mosquito LV or a Formulatrix Fast. All of these are liquid handlers. This is a liquid dispenser, and then these are liquid handlers moving things between plates. This is basically a automated 8-channel pipette with pipetting ranges from 1.2 microliters down to 100 nanoliters. This will do 13 microliters down to 100 nanoliters. Not shown here is the gigantic humidity chamber that you have to have around the deck to keep things from evaporating as you pipette 0.2 microliters around, and the plates sit for, you know, upwards of 30 minutes while this guy runs. You gotta keep the humidity chamber fogged up, otherwise things will just, you know, evaporate. And then you absolutely have to have an Eppendorf mixmate to keep things mixed, because this has a particularly small diameter of rotation. Okay, then once you get your plate set up, you're going to have to run a PCR on them. So you have to buy 2 Proflex thermocyclers, dual 384 Proflex thermocyclers. And then once you're done with the PCR, you have to pool the PCR products and clean them up. And so you can take your pick of a Hamilton Star, an Eppendorf EP Motion, or another Formulatrix Fast. These 3 are the officially supported robots by Olink. Agilent Bravo is unofficially, officially supported. So this is what we had. We had an NGS option A Bravo, so we actually had to pay Agilent $10,000 to come out and basically rip out all of the cool NGS stuff and take it back to a vanilla deck Bravo so that we could use it for Olink. And then we have to get another Eppendorf mixmate. Okay, so it costs what? So that's what we say when we tote up all of the CapEx that we have to expend. So an SPT LabTech Dragonfly is going to cost you about 60 grand. The Mosquito or the Fast, that's going to be another 120. You've got 2 Proflex dual 384s. That's going to be 9 grand a piece, or 18 grand total. And then you're going to need a post-PCR liquid handler. So one of the one of these four will run you somewhere between 80 to 120,000. So expect, if you want to start implementing Explore HT, you're going to spend somewhere between 300 and 350K in CapEx. So it costs what? This is not a cheap assay to run, and it's also very consumable heavy. Okay, so we need 384 well plates, dragonfly reservoirs and syringes, a whole bunch of fast tips, plate seals, a whole bunch of tips and tubes and strips and more tips and beads. And then you're going to need to run this on a NovaSeq X10B100 cycle, and they're going to need to pay the tech to run it all. And then the kit itself, list price, 60 grand. So you're looking, every time you run an Explore HT run, you're burning not quite $70,000. Now, any core director worth their salt is going to be able to get a pretty significant discount on this. But because discounts vary depending on how good of a haggler you are, I just put the list price there. So it's a lot of money, but you're going to be doing a lot of stuff on a lot of different samples all at the same time. So it works out to about $406 per sample for 172 samples run. Okay, artisanal batches. You run 172 samples at a time, or you run 86 samples at a time if you run a half reaction. So we have sample plate 1, we have sample plate 2. Each of these sample plates together in a 96 well format, they get combined together into what's called the sample source plate in alternating columns in the alternating quadrants. So this is important because, what is the matrix? Well, no one can be told what the matrix is, but you should be told what the matrix is because you need to know what your sample is that you're running, because you can't run every sample together in Olink Explore HT. So we have the standard run, so serum and plasma, that's the easiest possible thing you can do. So serum plasma, blister fluid, or bone marrow, those get the standard dilutions. So Olink Explore HT comes in 8. The assay is run in 8 different dilution blocks. So blocks 1 through 4 are neat, so we run the samples straight as they come, and then we do a 1 to 10, a 1 to 100, a 1 to 1,000, and a 1 to 100,000 dilution. And those are blocks 5, 6, 7, and 8 respectively. Now, the alternative, so alternative dilution, I think it's blocks 6, 7, and 8 aren't, or they don't go all the way out to the 1 to 100,000. But that's for CSF, tissue lysates, ocular fluids, blood spots, synovial fluid, and then we have neat. So this is where you only dilute out the controls. Everything else gets reacted as is with no dilution, and so that's for cell lysate, supernatant. So basically, you can think of this as what is the decreasing amount of protein in each of these matrices, requiring less and less dilution. Okay, so when you're running this, maybe a brisk walk. So day 1 takes about 1.5 to 3 hours, depending on whether or not you've got a mosquito or a fast. So 1.5 to 3 hours. Day 2, you're mainly just running PCR and loading the sequencer, and that takes 2 hours hands-on, 5 hours clock time. Day 3 is just 1 hour. So one pipeline can do anywhere from 688 to 1376 samples per week. This does not seq well with others. So custom recipes are required, mixture of read and dart cycles, and then you have to run their special program instead of the standard Illumina program. They have a really awesome built-in analysis program that lets you do a whole bunch of interesting quality control and other metric analysis without really having to do anything other than load the data in. Sometimes things don't go well. So here you can see a technician double loaded this column and skipped that column. And so this is the sort of thing that makes you go get a cup of coffee and take a walk around the block, because basically we've screwed up 16 different samples. And so we either have to rerun the whole plate or find somewhere to rerun those guys. And so that's why we bought an Integra 96, so that we could, instead of using an 8-channel, just do all 96 at one time for certain steps. So finally, synergies. So why is this awesome in a sequencing core? So the SPT LabTech Dragonfly is absolutely amazing when it comes to filling up 384 well plates. Sometimes we have to do full 384 well qPCRs. This will fill up a 384 well plate in 30 seconds with ultra low dead volume, so we're only losing about 30 microliters total. Small volumes mean small prices, so we've been able to miniaturize watchmaker RNA and DNA library preps down to 1/5 the reaction volume called for in the protocol, which makes our cost lower, which we can pass on to other people. Okay, and so what does it all mean? Basically, know what you're getting into. You're going to spend a whole lot of money to get here. It's a high wire act. You're going to spend a whole lot of money on a single-use only sudden death kit. So be very, very careful about what you're doing. Might as well use it. Basically, you're going to get a whole lot of really cool liquid handling stuff, and use it to do other things like miniaturize your other library preps. And then just to end it, in the musical Oklahoma, there's the farmer and the cowman should be friends. So I say the seeker and the mass spec should be friends. So what I'm doing is akin to RNA-seq, and what you're doing is akin to qPCR. We can still live amicably together. So, okay, so thank you to my crew, thank you to users across the world, thank you, and I'm done.
Sorry. Thank you very much. And, all right. Yes. Yeah, like I have a. Yeah, I guess I do not have a mouse. I'm not really gonna point to anything, so should be good to go. Hello, thank you very much for everyone being here. So my name is Sebastian Paez, and I work at Talis Bio as a data scientist. Just as context, my background is in open source development and with like a side gig of a cancer biologist that ended up doing computational mass spectrometry. So I guess I tend to like to know who I'm talking to, so let's see a show of hands. Who thinks of themselves as a proteomics practitioner that is AI curious in this room? Okay, right on. And what about the opposite? Kind of like who thinks of themselves as more of a bioinformatician that is more like proteomics adjacent? Cool, perfect. And who thinks of themselves as kind of like a pro in both? Right on, cool. Right, so like this talk is gonna be tailored mostly for the first group, so sorry if I was for the other ones. It's gonna be a little bit of review for you, but I'm happy to answer any questions that you might have. And hopefully we're gonna go through a couple of things, and my main idea is to give you like a more expansive view of like what artificial intelligence is and what it's doing in our proteomics workflows. And for the people who are more proteomics practitioners, for them to have like some basis to be able to like understand and evaluate some of their tools to see if like they're doing what they're expected to, and kind of like simplifying the process of like understanding what these tools are. So like for us to have like some shared understanding, like let's start with like what the definition of AI is, since it's a pretty overloaded term nowadays. So like I got this from this like startup company called IBM, and IBM defines artificial intelligence as like trying to emulate processes that like resemble human intelligence, right? Which is not a great definition because it can encompass nearly everything. But basically, that is what it's doing. It's like a series of tools that we use within like software development that try to incorporate what we would do as humans within our analytical workflows. So like if we think of like within software development, we're gonna have like a subsection that is gonna be artificial intelligence. And within this subsection that is trying to emulate human intelligence, we have machine learning, which is gonna be the core of this talk, which is not only we're trying to actively emulate human reasoning, but we're also allowing the program to learn by itself from the data that is presented to. And this can happen either at the time that you run the program with the data that you are giving it, or it happened before the program was like compiled and given to you as a user. And so it uses that prior information. What we're not gonna talk about is those two things that are gonna be in there. So deep learning is like a specific subsection of machine learning that doesn't really matter in this specific use case. And like for the generative AI, which is like the systems like ChatGPT that you might or might not have ever used, is also like a subsection of that. So just like to know where that falls within this like theoretical framework. So like whenever you're thinking about your like software without ML, you're gonna have like some sort of input, some process is gonna go down and it's gonna give you back some results. That can either be displaying something at the screen or giving you your actual count matrix or equivalent in your proteomics workflow. And the thing that is different whenever you're talking about software that actually incorporates machine learning is that it's gonna have some training step somewhere, right? Like there's gonna be a modification in your program that is gonna change its behavior, that is gonna be based on data and not like hard-coded parameters within the behavior of the software. And usually people think about this as like a magic box. Thanks, Ben, for this GIF. So the main idea here is like, how can we peel apart a little bit of this onion to like see what is important about this black box and how can we understand our tools a little bit better. So like at the end of the day, everything in machine learning is just number crunching from one type of number to another type of number. So whenever you're looking like what I like to call the three pillar evaluation system for like your machine learning workflows is like think of what is the data coming in, what is the data coming out, and by which process is it incorporating this data. So whenever you're like reading a paper and like looking at a machine learning, like it says like we used AI, kind of like go through the method section and like figure out exactly what aspects of it is it doing, and we're gonna do that with a couple of examples here. So like figure out what data is it using, how is it using it, and by more or less what mechanism is it using it. Like within the model section, you're gonna see a lot of specific lingo to the field that doesn't really matter. Like you could be, like you're gonna see things like deep neural networks, random forests, like support vector machines, and none of those are like implementation details, but what matters is like the data that comes in and out, and everything in the middle kind of like, unless you're actively developing machine learning, does not really change what the model is doing. All right, so like let's look at an example or workflow. Who has used FracPipe? Okay, cool. So first off, like whenever people tell me that they use FracPipe, like it doesn't give me any information. So we actually break apart what FracPipe is doing is that it's running for us a couple of steps. So like which of these steps use machine learning? I don't know. Which is the first step that you think uses machine learning within this workflow? Like does MStats use machine learning? Well, it doesn't. What about IonQuant? Does that use machine learning? Yes or no? It actually does. Does MS Booster use machine learning? Yes. It does. Does MS Fragger use machine learning? No. Good. Does MS Convert use machine learning? I cannot answer that because it uses vendor libraries and I don't know what those are doing. But what about like the instrument itself? Does that guy use machine learning? Yeah, it does actually. So like this is a patent from this other startup called Thermo. And like, what I want to show here is, like, obviously patents are not written for scientists, they're written for lawyers. But what we can extract from this patent is that it is actually using, like, as it acquires the data, the way that the automatic gain control is used, it is using, like, information from the last, like, 10 seconds that it has been running to basically figure out whether it is, like, on the rise or on the dip of the peak, and it's going to adjust your automatic gain control in that way. So, like, the reason why I'm showing this is because there might be a lot more machine learning that you realize there is in most of your proteomics. So, like, even before you look at the peaks, like what you would love to call your raw data, that's cute, but that's not raw. Like, it has been processed in so many ways before it gets to you, and sometimes you do have access to it and sometimes you don't. So, like, try to, like, be a little bit more inquisitive on exactly all of those things, because most of the times it's not gonna matter and you're gonna be able to use that correctly, but sometimes there are gonna be weirdness in your data that is explained by these internal machine learning systems whenever they don't work as expected. So like if we go back to this, like, 3 framework side of things, right? Like, we have some data, we have some mechanism, and we have, like, some model. Like, and we want to analyze ion quant, like, and we were to, like, break it apart. This is going to be figure 1 of their Alexei's paper, which is actually pretty good. And we can see that, like, the task at hand, I have some DDA data from 2 different samples. We have some peptides that are not identified in either of them. I would like to transfer the identification, but that I can do that in a rule-based system, right? I can just say, like, anything that is, like, less than 5 seconds away in the other run counts, right? And that will do the job, but, like, we can do a little bit better. What if we incorporate more data, right? Like, if I was a scientist, I would say, like, well, if the mass error is a lot higher in this other run, I would probably not consider it. If the offset of retention time is maybe a little too high, I would not consider it. And if I incorporate further information, we would give, like, a more broad view of, like, what we would require. And that is the process that is formalized internally within IonQuant. It basically derives kind of, like, what is the difference of retention times between my 2 runs, what is the difference in the isotope envelope distribution, and, like, a couple of the mass errors. So, like, just by looking at that, it can give you some information on what cases might or might not work for you. And in some cases, you're better off just, like, negating some of those features if your system is not something that matches, right? So, like, if you're doing a direct injection, probably your retention time information is hurting you to, like, be able to transfer your features if you're doing some kind of, like, wonky acquisition. So, like, all of those are really nice things to have, and that shift here and there. So, like, this guy in particular does not use external data for the training. It uses only your internal data to derive this, like, quote unquote probability, please don't kill me, Justin, for this one, of, like, it being, like, correct or a mismatch with your other sample. What about if we go into MS Booster? Like, this one's a little bit more complicated, because this one does use external data. Like, at the end of the day, what MS Booster is doing is that it is adding information to our search engine to give us, Justin is actually going to go into a lot more detail in this one, so I don't have to, but it's trying to give us more information on our scoring, more scores that we can incorporate to see whether our, like, identification is true or not. So if we had to put it in a more diagram kind of way, we have, like, the top section that is what the models that were trained before we give them our data, and the ones in the bottom are the things that are actually trained on the run whenever we run our program. So we can see on the left side that, like, we have a peptide, like, we are calculating the intensity correlation versus a theoretical spectra that is generated by my machine learning model, right? So, like, the first question is kind of like, what is it predicting it for, right? Because, like, not all instruments are the same. How does it figure out, like, that it is predicting? So, like, if you are running an HCD experiment and this model is predicting B and Y ions, like, will it work for you? Probably not. So that would be one of the things that I would, like, look for whenever you're processing that kind of data. Like, you would go into the paper, see, like, what their training data was, and derive, like, how is it representing. So, like, in this specific case, I believe at the time of writing the manuscript, it might have changed, they were predicting only BIY, and so this feature would be completely useless if you were doing an ETD experiment. On the other hand, it is, like, predicting quote-unquote peptide properties. And this is going to be your retention time and your mobility. And at the time of writing the manuscript, it was using, for the most part, like the big process library. So it's going to have like a mixture of immunopeptides and tryptic peptides. But like, if my samples have non-canonical amino acids, will it work? Like, you might be able to hack your search engine to like analyze that kind of stuff, but that feature is going to actively hurt you and make any non-canonical amino acids that you're running worse. So like, by understanding how it is using your amino acid information to derive your theoretical retention times, that might be like actively hurting you. And I'm going to show you an example of that later. So again, like if I had to like break apart like what is MS Booster, I'd say like nothing in the middle really matters. Like what matters to me is like it is predicting B and Y ions, it is predicting retention times, and it is trained on a mixture of tryptic and immunopeptides, right? So like if my data is not within those sections, it's probably not something that I would like to use. So just like know that nearly every step within the proteomics workflow is going to have like an AI component in it, whether you want it or not. And sometimes it's good to be a little bit more inquisitive on this kinds of data, in the same way that you as a scientist would evaluate your mass spectrometry system, right? Like I don't understand how like oscillations of voltages make a quadrupole work, but I know what a quadrupole does. And I would like people to have like that same high level of understanding whenever they're evaluating their tools. So like there are a couple of concepts whenever you're like evaluating a machine learning tool. For those who are not super familiar with it, like the first concept that I wanted to introduce was the concept of cross-validation, right? Which is you're gonna have like if I want to prove to you that my tool is good, like I want to show you that it like matches the reality of some data, right? The trick here is that it has to be data that it has not seen before, because complex models are surprisingly good at like memorizing your data, and you have to be able to evaluate it with something that is like close but not quite what it has seen. So the way that that's usually done is that you have like some big corpus of data, like let's say I have like 200 runs, like I would train my model in like the first couple of hundred and then evaluate in like the others. The big issue, and something that kind of like the big no-nos whenever you're like reading a paper and seeing whether when they're trying to prove that their tool is good, is whenever they double dip, right? Like they're using the same data that they trained on to evaluate their model. And that's like, it happens a lot more than you think it should. And it happens sometimes in very complex ways. So just like make sure that you at least look for any presence of that or a mention of how they alleviated that from happening. So when someone tells you, we randomize samples, we run the same ELISA 20 times, and it turns out that if I split randomly between my runs, like my prediction for retention time is absolutely outstanding, right? Like I would at least like to see samples run in another instrument in like another lab that is going to be like at least not that batch specific to know whether this tool is going to generalize to what I need it to be. Another issue that sometimes happens, but this is more on the user side of things that can happen accidentally, and user being me, it is that you might have data that is like out of the distribution of the sample that I have seen before, right? So like similar to what we talked about, the HCD example, like if your spectrum predictor has only seen data from HCD from Orbitraps at collision energy 27, probably if you're using a science instrument, you might not have the best results, right? So like always like look at whether the data that the model, or like the underlying engine that is being used within your tool is using is like within the distribution of what you need. Because again, like you want it to be not exactly the same data that it was trained on, but you want it to be close enough. Like if I train something that is classifying between apples and tomatoes, if I give it an orange, it might do a good job, but if I give it a papaya, it might do a bad job, right? Like sometimes, I don't know. That's a horrendous example, sorry about that. And then like whenever you're making informed decisions, sometimes some labs are a lot better about this than others. Some of them will give you like a lot more information. So if we look, for instance, at the Prosit paper, it tells you like in the first version of them, they used 500,000 tryptic peptides, right? So we have an expectation that is going to work beautifully for tryptic peptides and not that great for much other things, right? Which they acknowledge in their second paper when they say like, hey, by the way, this sucked at immunopeptides, so we like now did a bunch of immunoruns. And if you look at the data from their second paper, and especially in the left side, you basically have a doubling in the quality of the predictions just because they actually use data within the distribution of what they needed it to be. Yeah, so like if you're using like any of these models, kind of like if you have like a non-model organism or something that has weird amino acids in it or something like that, it is really worth like looking on whether that still fits your assumptions of it being like within the distribution of a canonical amino acid. Which brings us to this awkward point to talk about, because like some tools don't give you this information, right? So like my general recommendation is trust but verify. Like in general, I prefer open source tools because you can actually look at, if you know how to write code, or if you have a friend that knows how to write code, they can actually go in there and see whether what they described in the manuscript is actually happening. And if you cannot do that, sometimes you can run the tools in specific ways to trick them to see whether they are actually doing what they're meant to do. So always try to use software that is open in my experience. And if you cannot, try to ask the authors how they're doing that kind of stuff, if it's available. And if they don't provide any of those things, run. So talking about how things can break down whenever you're using machine learning in some of your tools, I actually wanted to give you an example. This is a lot of text, but you can ignore it and just listen. I had like some samples where I had like some cells and we were treating it with a compound called NMIAA, which is a variant of like your iodosetamide that you would use for your sample prep. Which basically meant like, if I treat my cells, any cysteine that's exposed is gonna get modified with 71. Then whenever I carbamidomethylate it, like any cysteine that is not exposed is gonna be modified with 57. I run a first search, did a standard variable modification, like looked gorgeous. A couple of hundred peptides in each of those two different components. But then like my lab mates were kind of like, we think like this guy might be getting a different adduct. Can you run an open search for me and see whether it works out? And whenever I run the open search, like basically nothing. Like absolutely every carbamidomethylation and modification disappeared. So like what could have happened here? So like, since I knew my tools, one of the things that like I knew was like Percolator uses very heavily like the difference in masses that you would have between like a theoretical peptide and like the observed one that you would have. And whenever I looked at the data, it was getting reported kind of like this. So like in my closed search, it was saying like I have a beautiful mass error of 3.1, but in the open search, it was reporting my error with respect to the unmodified peptide. So I had a mass error of 57,000 Daltons, even though it was actually pretty tied with the respective one. So like just being able to do the forensics of like knowing that my tool is looking for a specific feature and then looking at whether my raw data has the same specific feature and do they match has been like extremely powerful for me to like understand what my tool is doing. And so like more conceptually speaking, what was happening was like the way that my search engine was reporting stuff was generating out of distribution data, because like there's no way that the model could have known that there's like this weird offset that it actually matters. And I wanted to end this talk kind of like talking about speculating a little bit, right? Because like the hardest thing to predict is always the future. And one of the things that I feel like is happening in the field, and I think it's gonna continue to happen, is that ML is gonna expand in its scope. So like right now, the most common applications that we have, which is nearly everything that I showed, is like once your search engine is done searching, how can we augment the data by using tools that actually incorporate machine learning? What I see happening a lot is that the machine learning layer is getting closer and closer to the search engine to be like a critical part of the functioning of the search engine itself. And an example of that is this tool that Villanoble's lab wrote called Casanovo, which actually uses like machine learning as it searches your peptide to derive like de novo sequences. By the way, it's a great tool if you want to try it out. Conflict of interest. One of the authors is my boss, so. But like those are not that's not the only one. Like if you look at some recently published paper, like there's at least 5 or 6 different companies and/or tools that are doing either direct scoring using machine learning or are pushing the machine learning closer to the search engine layer. So if you're a Proteome Discovery user, Chimeris is an example of one of that. If you would like to hire a service provider, Tesori, I think it's doing a pretty interesting job. And AlphaDA from the Man Lab also does that directly. So that's like a big breaking point from like the status quo of how like search engines work. I think we still have questions on how to evaluate those tools efficiently. But I think like that's definitely a trend that I see continuing. I also see kind of like once you get that like weird Excel table with samples on each column and peptides on each row, kind of like what do you do with it? I can definitely see some insights happening. And just to show that it's not total hype, like Google just like released now, so if you go into Google Colab, you can drag and drop the results of your search engine, say, this is proteomics data, can you analyze it for me, and click enter. And it does a pretty darn decent job. Like I tried it this morning while I was procrastinating, rehearsing this talk. And it did, for the most part, everything that I would do for a general exploratory data analysis. And the crazy part is like, this is the worst model that you will ever use, right? Because things are just gonna get better from here. So kind of like, I definitely see this like being a critical part of the field and evolving into things that are custom to our specific needs. Like right now, it didn't do it for me a pathway enrichment analysis. It did not do for me anything that would like look at the raw data. But I can see how like a smarter system that has access to those tools would be incredibly valuable to assess some of these. Right? Like you can imagine being able to type kind of like which of these peptides actually have a chromatogram that does not look horrendous, which is a question that some of us have, but like evaluating on a large scale is pretty hard. And I guess like on a provocative last slide, wanted to see kind of like there are fields of proteomics that are definitely underrepresented within AI. So like I have not seen a lot of development neither in top down or middle down or any of other wonkier variants. Or like in even like non-model organisms tend to be not all that well predicted. Or non-canonical modifications seem to be a thing that has been problematic historically. And that is like, I do not see it getting faster despite how much I would like it to. And also like, I would like to, like, AI has this weird effect where it's kind of the rich get richer kind of deal, because like the more data that you have to train your model, the more better it's gonna work on that specific kind of data. So if everyone is running Orbitraps with HCD, like all the models are gonna work great for that. So all our tools are gonna be good at working with that type of data, but that could be hindering the development of new fragmentation methods, for instance. So like keeping an open mind and like supportive of that development of like new technologies could actually be hindered depending on like how widely adopted our AI is. And obviously like things like I have not seen any model that predicts correctly like non-C18 based chromatography, right? Like it like permeates into like other fields as well. So yeah, again, like I just want you to, again, the takeaway from this talk is be a little bit more inquisitive of these tools. You don't have to understand every single detail. Know which numbers come in, know how does it, kind of what it's looking for, what numbers come out, and ask yourself kind of like, within my tool, how is that working? Like the same way that you don't have to understand voltages in a quadrupole to know that it's filtering ions. If you want to keep learning, I'd say if you're a PI, focus on the left side. If you're a trainee, focus on the right side. I think like it's incredibly valuable. And conflict of interest, again, Will Foundry is my boss. He gives a seminar every year at ASMS about mass spectrometry and machine learning. So thank you very much. No questions right now because
Hey everybody. Thank you, Sebastian, for kind of like that great overview and kind of sets me up nicely. My name is Justin. I'm a graduate student in the Noble Lab at the University of Washington. My lab works on a number of different machine learning and deep learning tools for proteomics, but today I'm going to be kind of throwing it back a little bit and talking about one of the earliest ML methods for proteomics, Percolator, which was developed by one of the predecessors in my lab a very long time ago, at least from my perspective. And I think Percolator is a very useful algorithm to talk about as an introduction to AI and proteomics, both because it is like a very good vignette for showing like how you need to be careful to do kind of AI correctly and how you can easily fall into pitfalls which would lead you to kind of totally break your analysis pipeline and maybe mislead yourself and get unconvincing results. And also, many of the new AI tools in proteomics kind of build into the Percolator framework, and it's kind of the foundation that they're all kind of sitting atop of. So for people who are already an expert in Percolator, I apologize, this might be a little bit of review for you. But kind of starting with a general background, because I think people here are from very different backgrounds. In a mass spec experiment, we've digested a sample into peptides and acquired tandem mass spectra, where we observe a fragmentation spectrum for each peptide. And our goal is to figure out what the generating peptide of all of these observed mass spectra are. And the way that this is traditionally done in the field is with database search, where we take the reference proteome of the sample that we're analyzing and digest it in silico into all the possible peptides that might have been present there, and then look at kind of the candidate peptides which could have generated this spectrum, and compute for each of those peptides some theoretical spectrum where in like an ideal universe where I saw perfect fragmentation and the fragments are super abundant, what would the spectrum for that peptide look like? And then I compare that to the observed spectrum using some score function. Some of the most popular ones are XScore, which was introduced by the original Sequest search engine, Hyperscore, which is implemented in MS Fragger, but there's a bunch of different score functions. They all, at a very high level, look at kind of broadly like how many of the fragment ions that we would expect to see are present in the spectrum, and how much of the intensity in the spectrum, how much of the signal comes from the peptide. And these kind of give a kind of similarity score, assessing the quality of the match, and we'll take the top matching peptide for each spectrum to get a peptide spectrum match, which I refer to as a PSM. And looking at a couple of individual PSMs, here's a peptide that's very close to my heart. And this example looks like a very good PSM to me. Many of the theoretical fragment ions for the peptide are present. They're very high abundance. Most of the intensity in the spectrum is explained by fragments of this peptide, and this corresponds to an XScore score of 2.8, which I guess maybe looking at this is a pretty good XScore score. On the other hand, here is a kind of what I would think of as a pretty low quality PSM. There's only a handful of theoretical fragment ions present in the spectrum. They don't explain much of the total signal, and correspondingly, there's a very low XScore score of 0.4. So based on this, I might say this is an incorrect PSM. Sanders is not the generating peptide for the spectrum, rather it's the result of a random match between the spectrum and the database. And this is kind of incorrect. So obviously I can't go through my experiment and look at each of my PSMs one by one. There's tens of thousands of them. And so in order to make this scalable, we need some like automatic way of assessing which are correct PSMs. If I take all of my PSMs for the whole data set and look at the distribution of scores, for the XScore score, I would see something that looks like this, where way out on the right, the highest scoring things, heuristically, I'm very confident they're correct. They are very beautiful spectra, really good match. On the other hand, the lowest scoring things, way over in the left here, are pretty crappy PSMs. They are almost certainly an incorrect identification and the result of just random matching to the database. But where exactly the threshold I should kind of set in this score distribution is a little bit unclear to me. We know it's a mixture between kind of a null distribution of random matches and an alternative distribution of correct matches, but exactly what those mixture components are is kind of complicated. And ideally, I'd like to be able to pick a score, take everything higher than that score, and run with that for my downstream analysis. And the strategy that the field has adopted to do this is called target decoy competition, where during the database search setup, we add an additional decoy database of random peptides, usually obtained by like shuffling or reversing the protein sequence in your database. And then when we are searching an observed spectrum, we search it not only against the target database, but also against this decoy database. And then we take this top scoring match between the two databases to obtain our top PSM for each spectrum. And this is really useful because for a given observed spectrum, if we have a correct identification, it is definitely going to be from the target database because none of those decoys were truly present in our sample. But if we have an incorrect identification, and this is just a random match, it's equally likely to come from the target database as the decoy database. So if I look at the distribution of scores assigned to decoy PSMs, I get a really good estimate of the null distribution of kind of random match scores. So I can estimate one of the components of this top mixture distribution. And since we know that for kind of every incorrect identification, there'll be one decoy and one target, I can kind of look at the overlap of these two distributions and say at some score threshold, if 1% of all of the IDs above that score threshold come from decoys, there'll be a corresponding 1% of the targets which are incorrect, and this allows me to assign some statistical confidence to my discoveries. Which is wonderful. I can now kind of take these IDs and move forward in my analysis. But looking at this distribution, you might be thinking to yourself, maybe we're kind of like missing some stuff here. Like X-score doesn't look like it's the most useful score function it could possibly be. There's a lot of like IDs here, which there's overlap between the alternative and null distribution, and if we had a better score function which separated those two distributions more distinctly, for example, some hypothetical score function that looked like this, we would improve the power of our database search and be able to kind of return more identifications to the user. So how could we obtain such a score function? Well, you might think to yourself, maybe I should use some of the additional variables that I know about a PSM, which might be indicative of how likely it is to be correct versus incorrect. For example, if I look at the length of a peptide, I'll quickly see that X-score scores don't look very well calibrated with respect to length. The distribution of scores assigned to very short peptides, there's way more overlap and all of the scores skew shorter just because there's fewer total fragment ions, so the highest X-score score you can possibly get is lower. There's less stuff to match. Looking at longer peptides, we see that the separation of target decoys gets better and the scores in general shift higher. And then for the longest peptides, this becomes even more pronounced. So looking at this, I might think to myself, well, I could, instead of just picking one threshold in the score distribution, I could pick 3 different thresholds. I start by like my length 8 threshold at a 1% FDR there will be a little bit different than my length 8 to 16 threshold and my length 16 plus threshold. And by including this additional length variable, I can slightly increase the power of my database search by fixing this miscalibration. Wonderful. Let's apply that same idea to kind of all the other variables. I'll quickly run into a problem where there's a lot of different variables that we have access to which might be indicative of PSM quality, such as the length that we just talked about, but also the charge of the precursor, whether or not this peptide is the result of tryptic digestion, the number of internal miscleavages, the mass error between the theoretical mass and the precursor mass, and so on and so forth. And picking a different threshold for each possible combination of these blows up combinatorially. So ideally what I'd like is kind of some model which can synthesize the information from all of these variables and what they're telling us about whether a PSM is likely correct or incorrect, and combine them into kind of a single combined score which we can hopefully is better than each of the individual variables themselves. And this is exactly what the Percolator algorithm aims to solve. So Percolator is a tool which re-ranks PSMs based on kind of a list of available features. It takes in kind of a list of all of the PSMs from your data set, whether or not they're a target decoy, their corresponding score, and whatever other features you want to provide about them. It uses a machine learning algorithm, which we'll go into much more detail coming up, and then it returns kind of a re-ranked list of identifications where it's combined all the information it has about each PSM to hopefully better separate targets and decoys than just the score function would. And then it also does a second thing on top, which is it performs FDR control on the new list. So it'll draw kind of a threshold on that new score and tell you everything above this threshold has whatever your requested false discovery rate is. So how does this work? Well, Percolator trains an SVM to separate correct and incorrect identifications. This is kind of a very common misconception and an abuse of like terminology. People often say we're separating targets and decoys, and that's not quite right. We'll discuss more, but like if you were separating targets and decoys, that wouldn't be useful. We're using the decoys as kind of a surrogate for the incorrect targets. And what we really want to be able to do is separate the correct target identifications from the incorrect target identifications. Percolator uses an algorithm called a support vector machine to do this. And an SVM really is just extending that idea of drawing a line in the histogram, which separates it into kind of two halves, to include multiple variables. So here is a situation where we have added an additional variable to the data. We can't visualize more than 2 dimensions because it starts getting very complicated, but you can think of this as here on the x-axis is the X-core score, on the y-axis is a second feature, for example, length. And now we're going to draw our threshold not just on one of these variables, but kind of a combined threshold on 2 of those variables to separate correct and incorrect PSMs. And each of the points here corresponds to a PSM colored by whether it's a target or a decoy. So how does SVM work? I'm going to give a quick little non-mathy aside here, just to have some extra ML content for people who are really looking for that here. The way an SVM works is it's an algorithm for performing a classification problem. So here's a little toy example where we have patients with either a disease or controls, and we're trying to see whether we can classify the disease based on 2 biomarkers. And obviously these 2 kind of samples are clearly separable from each other, but there's many different decision boundaries we could pick to say like where is our line that separates disease and controls. We could use this, and all of these perfectly separate the 2 classes, which is quote unquote the right one. And each of these kind of lines here in the context of SVMs are referred to as a hyperplane. A hyperplane is a fancy word for kind of a surface which splits space in 2. So in 2 dimensions, it's a line. In 3 dimensions, it's a plane. In higher dimensions, it's a hyperplane. The core idea of an SVM is that the best hyperplane to use as a decision boundary is the one which maximizes the margin, which means it's the furthest away from points of either class. And this kind of makes intuitive sense for how I would pick a line to draw here, because it means it's going to be the most robust to kind of additional noise in the data. As I kind of sample future points in my test set that I want to apply it to, I want those to be kind of least likely to accidentally cross the line of our decision boundary, so we want to give them the biggest buffer as possible. And then one kind of minor flaw with the way I've been presenting it so far is that oftentimes we have really noisy data. There are confounders. It's not possible to perfectly separate our data. And to accommodate this, SVMs allow you to relax this constraint of saying, okay, we'll have a penalty for when you misclassify a sample. And then given kind of a given number of misclassifications, I also have an incentive for maximizing the margin. And you can weight this trade-off between more misclassifications but a wider, more robust margin versus don't allow any misclassifications but maybe have a decision boundary which is super sensitive to very minor changes in the data. And how you decide to kind of weight those 2 trade-offs is a hyperparameter that you have to set when designing your algorithm. And a hyperparameter is sort of just a modeling decision is made when the algorithm is initialized, which is going to affect what the kind of final results of your algorithm look like when you apply it in practice. All right, so that little vignette aside, let's return to kind of how we use an SVM in the Percolator setting. You'll notice here that the kind of setup is a little bit different from that kind of biomarker problem before, where we had labeled positive disease samples and negative control samples. Here we have labeled ground truth negatives, in the form of decoys. We know that every decoy PSM is an incorrect match, but we don't have ground truth labels for positive examples. We have target PSMs, but those are a mixture of correct and incorrect identifications. So this is called a semi-supervised learning task in machine learning. It means that we only have labels for some of our data, and more additional set of our data is unlabeled. And the way that Percolator addresses this is by kind of inferring a noisy best guess at the labels to start, and then iteratively improving the labels it's assigned. So we start by kind of doing exactly what I described previously of calling PSMs at a 1% FDR using like a threshold on this X-core score here. And then saying that all of the identifications above that threshold are correct, and we'll label those as the positive class, recognizing that 1% of those are actually incorrect. But that's not a huge problem because these machine learning algorithms are fairly robust to a little bit of noise in the labels. So even if 1% of our labels are wrong, that's okay, and it's going to be able to kind of accommodate that fact. And then our negative examples are the decoys. And then based on these label assignments, we'll kind of use the SVM algorithm to learn a decision boundary, which best separates targets and decoys. And then using this new and improved decision boundary, you can see now we're taking the second variable X into account a little bit. We'll recall identifications at a 1% FDR. This is giving us a better labeling than our initial best guess. And then we can retrain the algorithm and keep on iterating back and forth between kind of make a better decision boundary, use that improved decision boundary to improve our labels, and keep going back and forth until the algorithm kind of converges, the boundary stops changing, and then we can finally kind of keep this decision boundary as like a weight of how important X is versus X score, so like length versus X score. You can see it's using both of those features in how it decides to separate correct and incorrect PSMs, and then we can draw 1% FBDR on that final boundary and return the set of targets which pass that threshold as discoveries from Percolator. And in practice, this works really well, depending on the data set and exactly kind of what additional features you're providing. Percolator tends to pretty dramatically increase the total number of PSMs identified in a data set, and it really, I think these days, there's not a reason not to use it. You just have to think about which features exactly you want to provide. But it's important to ask this question of like, what makes this algorithm valid? How do I know that these additional discoveries at a given FDR are coming about from improving the power of database search rather than I broke my FDR control and I'm totally misleading myself? And there's kind of really cool two key tenets for like why the Percolator algorithm works. The first is that we use cross-validation to keep the training and test sets disjoint. So this means that we start by splitting the data, all of our PSMs, into 3 separate sets at random, and then we use 2/3 of the data to train the algorithm, as I just described, and then we apply that learned model to the 3rd set as the test set. And then we repeat that whole procedure, learning 3 different models with very slightly different decision boundaries 3 times to make inference on each of the 3 test sets. And this ensures that a model is never trained on the same data it's being applied to, which prevents any concerns of kind of overfitting or memorizing the training examples. The second, I think, slightly more nuanced like aspect of Percolator which makes it valid, or assumption that it's making, is that decoy PSMs and incorrect targets are indistinguishable from each other. We, right at the start, made this assumption that kind of the decoy matches were a good estimate of the null distribution of random matches amongst the targets to the database. And that means that there's kind of one match to the decoys for every incorrect target, and the scores of those matches should be indistinguishable from each other. And that means everything's going to work as intended. But if we added another feature to Percolator, which was able to trivially separate all of the targets, both correct and incorrect identifications from the decoys, you could think of that being something like, I don't know, you did homology, you look for homology to other species where you're going to see really close matches for your targets, but there's random matches for the decoys, and you added that as a feature to Percolator, you'll get this kind of trivial separation between the 2 classes, where the decoys have a different distribution in some of the variables than the targets do. And that will let Percolator find this decision boundary which separates the decoys from all the targets, including the incorrect ones, and our FDR control will become totally broken. So it's very important to think about, like, as you're adding, like Percolator used, the original implementation used a pretty small set of kind of features, like the masked error, the whether or not they're triptych termini, which like they showed that this assumption is pretty reasonable for those. But as we start to add more complex features to Percolator, we have to think about whether this kind of remains true. And as I'm talking about more kind of complex features, recently these features are becoming kind of AI-derived themselves. And many of the recent kind of more recent AI models that you've maybe read about are kind of being used to generate better features for Percolator. That's how they're providing their value in the AI search program. So a very common one now is using kind of models which are able to predict the retention time for an analyte. So here's an example from, I think, DeepRT. There's a very good match between, like, very accurately able to predict when a peptide is going to elute, given just the peptide sequence. And so we can pass in the difference between the observed and the predicted retention time for a given PSM, and a small difference is very indicative of a correct identification. Incorrect identifications will have basically kind of a random, uniformly random difference. So Percolator weights this feature very heavily when it's doing rescoring. Another example is looking at similarity to a spectral library. I think early on this might have been like an observed spectrum versus some kind of purified library spectrum or consensus spectrum for a given peptide. Although these days it's very common that this is now an AI predicted spectrum where you've used a model like ProCit to predict the fragment intensities for each of the theoretical fragment ions, and you look at the similarity of that to the observed spectrum and pass that similarity in as another feature to Percolator. So now it's kind of AI inception. There's AI tools generating the features which are being passed to other AI tools. And it's very important to think about now whether these algorithms are still obeying the core tenets that kind of made Percolator work in the first place. Namely, it's something we see very commonly is that these models have overfit to the training data, and say it was like trained on human data, and you're applying it to human data, your retention time model might make better predictions for your target peptides than they do for decoy peptides, because it's never seen those decoy peptides before. And now you're starting to violate that assumption that the decoys and the incorrect targets are drawn from the same distribution, because you now have this retention time feature where there's even a very minor difference between those two distributions can subtly but very like incepidly break your FDR control in ways that you might not notice at all. And kind of now taking a step back and thinking about where the field is today, kind of almost every single pipeline that you're familiar with uses a combination of these AI tools. It gets a little bit confusing to understand exactly what's being done, because each individual step in the pipeline, which almost always includes now a spectrum or library prediction step, an RPT prediction step, and a CCS on mobility step, assuming that that's relevant for your data, are being used. Those models each have a name. They're then being passed in as inputs to, you can see this final kind of table here, rescoring algorithm, which is today still mostly Percolator, but occasionally there's some other algorithms which are ostensibly the same idea as Percolator, maybe just swapping out a different model rather than an SVM in there, or something that they think runs a little bit faster. But really, this column hasn't changed that much, but the features which are being passed into rescoring does keep on changing. And so kind of keeping track of how to, like exactly what is being passed into Percolator in your pipeline, whether it's making any assumptions which are going to break the rescoring, is a really kind of useful thing to think about when you're running your own analysis, and always be a little skeptical of kind of a new algorithm which hopes to pass in another feature to Percolator. You should think about, is that new feature going to satisfy all of the assumptions that Percolator requires? With that, I want to thank you for your attention. And open up to any questions from you or Sebastian. it's always a peptide, right? Where if it is a peptide, we've got a protein sequence. Why would the score distribution matter if, why reach out to non-models when it's, right? I mean, thinking it's just a peptide, right? So it wouldn't matter if the black widow spikes or whatever.
Yeah, so like, the distribution of amino acids between organisms is slightly different, and also depending on like their internal metabolism, right? Like they would have different internal enzymatic activity. So like sometimes the distribution of the lengths between like what is tryptic between species might be changing a little bit. So like it depends a little on like what exactly your organism you're talking about. So like if you're, I don't know, doing like like some like weird hamster versus a mouse, it's probably not that different. But if you're working with like an archaea that has like some really weird resistance to trypsin, probably most spectrum predictors are not going to do a great job at.
Okay, so it's more of a concern that it might not work well than, because we don't know, than a concern that it is not working well.
Yes, so like I would probably like, what I would do would be to try to like run it in the base way, kind of like on your search engine without any rescoring that would use AI, and see whether on the equivalent sample for your canonical organism for which it does work, that your distributions of amino acids are kind of similar at least. And if that's not true, I would do something about it, like, or not use that specific feature. So I don't know.
Okay, yeah.
Does that make sense?
Yeah, that helps. Thank you. No, okay, for you, sorry. Okay.
Thank you very much. It just occurred to me that there could be biological reasons that, especially for retention time prediction and behavior, for example, if there are D amino acids instead of L. And I'm wondering if there would be a way to use the so-called incorrect hits to see if they really have some biological relevance.
Yeah, so I definitely think one of the downsides that Sebastian mentioned of incorporating some of these models is it'll stop you from finding new and weird stuff. For example, like there's a subset of PTMs which none of these RT models handle, and they all handle a different subset. And oftentimes you'll find that like if they're really bad at, say, phospho, and they're predicting terrible retention times from that, you'll analyze your data and see no phosphorylation, and I say, what's happening? And you've kind of restricted your, like, they'll tell you a clue that there's something missing due to the fact that there's all these things which were originally scoring really high, and we were identifying them, and then after rescoring kind of fell off and disappeared. So they can definitely kind of give a little clue towards there's something I need to follow up on in my data. But oftentimes that things aren't working as intended, I'm not seeing the things I should, then needs a manual follow-up of look at some spectra and see exactly what's going wrong. Yeah, so like just as an amendment to that one, like just like know whenever you're getting your FDR from Percolator is giving you it is not the quote unquote probability of it being true. It would be kind of like under some assumptions, how often would you expect it to be randomly that high, right? So there are even like mass spec reasons for which you can have a very highly scoring peptide that is wrong, right? Like you can have a deamidation, you can have like in-source fragmentation, stuff like that, that it is not accounted for within Percolator. So like even though like what we would like to know is like how likely is our disidentification to be correct, like what we're actually getting is like what is this expected probability of it being random. So which are like two slightly different things that sometimes really matter.
Hi, David Butcher from the National Mag Lab. I just wanted to say thanks for mentioning top-down proteomics. It's the first time I've heard it mentioned at all at ABRF, and it's what I do exclusively, so thank you for that. I had a question related to the data leakage issue that you mentioned, I think, with the training data sets. So when we're training the model on a huge number of data sets, say they all come from the same species, then obviously we're observing the same proteoforms or the same peptides over and over and over again. Is the implication that we should reserve the same peptides from every data set to use for validation later? Or
Yeah, so I guess it depends. There are like a couple of ways of doing it. So like whenever I've trained models to predict peptide fragmentation to not trick myself into believing that it's actually working well, what I do is that I would isolate mass ranges. So like I separate, like even if I have 20,000 mass spec files, I don't separate them by PSM. I separate them by the mass of the unmodified version of that peptide, right? So I don't even use like an oxidized versus a non-oxidized in different compartments because I feel like that leaks data as well. And being a little bit strict about that is really important. I think that's also how the Wilhelm lab does it, and so far it's worked reasonably well, but just like know that it is like significantly better at predicting dose. Even if you read at the Prosit paper, like they would split it at the peptide level, and you can see that it's like a number that doesn't mean anything, but you would get like a correlation on your test set of like 9.9, but a correlation on your testing set of like 8.9, right? So they're like, there's definitely some level of like overfitting and like memorization of the examples that it has seen before that you would have that. So yeah, like being strict about like separating it and preventing ways that even if they're like very similar, that they're not like dipping in each other's toes.
Right. Since we're training on top-down data, our peptides are much, much larger.
Yeah.
So, the whole mass range thing is a little bit more of an issue because it's so wide. But
Yeah, so I think that's an interesting one because, like, I mean, there are instances where you do not have enough data to do like a full cross-validation, in which case I'd be very careful about like how you evaluate your model because, like, again, like machine learning is like the more complex your model gets, it gets surprisingly good at like memorizing some of the data that you have seen, and with that, which is why you have a testing set. Because like usually you would, if you have ever trained one of those, and you've probably seen the curve, right? Like where you see like the performance over time of training, and you see that it like starts getting progressively better. And if you only had a training set, it's only going to get progressively better until it memorizes all of the data in the training set. But then if you have a testing set, there's like a point where it's like memorizing so much that it's actually getting worse on like anything that it has not seen before. Yeah, so like some of that, it's a good thing to look for in papers. Very few authors publish those, at least in proteomics that I have seen, so.
okay, thank you.
Yeah. Let me add a point over there. So like one of the sources of data leakage that I have seen, it's not exclusive, like not exactly within proteomics, but it's on the field of like X-ray crystallography, would be that people claim like, oh, we have like this amazing performance on our X-ray structures because we downloaded all of the protein data bank and then split it randomly 75-25. But they did not deduplicate protein families, right? So it's kind of like if you have in the test set like a lot of the same family that you would have in your training set, like you're basically memorizing the same protein because like the chunk that crystallizes is nearly identical. So that is kind of like one of those like tricky times where you have like unknown data leakage.